Dr. Chuck Sanders, who's a professor of biochemistry and the vice dean for basic sciences. And he's going to tell us about targeting PMP22.
Good morning, everyone. It's such a pleasure to be here. And I want to thank the organizers. I know it's a lot of work to put a meeting like this together, and I appreciate all that hard work. So I'm going to tell you about a project that started 25 years ago. So 25 years ago, I'm a biochemist, biophysicist. I was looking for a membrane protein that was linked to human disease to study. It was as simple as that. And I wanted a protein that there was not already a community of people studying. So I stumbled on peripheral myelin protein 22. I had never heard of Charcot-Marie Tooth disease. No one else was studying at the sort of biophysical, biochemical level, so it seemed perfect. And so what was known even then was that a mutation at any one of these blue sites causes Charcot-Marie Tooth disease. And we now appreciate that these mutations cause the type E phenotype of Charcot-Marie Tooth disease. And so I decided we would try to figure out, how do these mutations cause the disease? And so our working hypothesis is that what these mutations do is they destabilize the protein, and it's this destabilization of the protein that ultimately leads to the disease. What do I mean by destabilization? So when a protein folds, there will always be an equilibrium between the folded form and the unfolded form. For a stable protein at the equilibrium, the folded form is preferred over the unfolded form. Okay, if you destabilize the protein, then the equilibrium shifts towards the unfolded state. And that's a problem because the unfolded form of the protein cannot do its function. And it's also very susceptible to either being degraded, in which case it definitely can't do its function, or forming aggregates, which may be toxic. Okay, so we propose that these mutations do exactly this. It shifts the equilibrium towards the unfolded state. So it took us 15 years to test this hypothesis. And so in 2015, we published a paper, and this is my postdoc, John Schleyback, down on the faculty at Purdue University, who did this work. And so he measured the stability of a series of disease mutants of PMP22. And so the red mutants are severe disease forms. These are intermediate disease forms. Light blue is a very mild disease form. And of course, these are our control here. And so here's the stability. And you can see the severe disease forms are the most destabilized. The very mild disease is basically has wild type like stability. And what we're plotting here on the y-axis is the cell surface trafficking efficiency. So for PMP22 to do its job, it has to make it out to the cell surface so that it can get into myelin. And so John also measured the trafficking efficiency. And you see there's a linear relationship between stability and the trafficking efficiency. So if you destabilize the protein will never make it out of the cell, out to the cell surface where it can do its job. We also collected patient nerve conduction velocities from the literature and plotted those versus stability. And once again, you can see a linear relationship that if you have severely destabilized protein, you get very, very low nerve conduction velocities and linear all the way up to wild type like conditions. And so based on this, then, for the type E form of the disease, we believe we understand the fundamental underlying cause of the disease that is caused by the destabilizing of the protein that causes misfolding, which leads to mistrafficking, which ultimately leads then to dysmyelination and peripheral neuropathy. So we were obviously very pleased with these results. However, how does this relate to Charcot-Marie Tooth disease writ large? Well, so the mutations that we're talking about are thought to be a relatively rare cause of Charcot-Marie Tooth disease. And so here is the pie chart. This is a bit dated, so we don't all have all 100 genes listed here, but the overall shape of the pie chart hasn't changed. And so the type E mutations are actually a relatively rare form of Charcot-Marie Tooth disease. However, if you look at the pie chart, the biggest pieces of pie are also associated with PMP22. either caused by duplication of the wild type gene, so you get 3 wild type alleles, or the HNPP form of the disease, where you are missing one of the 2 alleles. So in this case, you're making too much protein. In this case, you're making too little protein. So it's a long-standing paradigm in disease research that rare disease mutations can very often inform on the mechanisms of the much more common forms of the disease. And so we wondered, what did we learn about the type E disease that could be applied to the more common forms of the disease that affect the greatest numbers of patients? And I just want to make a point here, a rah-rah for Charcot-Marie-Tooth disease research. I mean, we did an analysis a couple of years ago, and it's very clear Charcot-Marie-Tooth disease as a whole is a top 10 genetic disorder. And yet most people have never heard of it. So that's why the work of the Research Foundation, the CMT Association is really so important to just get the word out that this is a really common disease. Anyway, so we set out to try to figure out what could we learn about these forms of the disease from what we had learned here. And so this leads to very recent work then. And so Mason here is here at the meeting and has a poster. And this is a postdoc from Bruce Carter's lab, Pramod. And what they developed was a Schwann cell line, a rat Schwann cell line expressing human PMP22 under the control of an inducible promoter. And what this allows us then to do is to simulate both the HNPP form of the disease and the common type 1A form of the disease by tuning the expression level of the protein in cells. And so what's going on here is we're adding a chemical agent that causes PMP22 to be made. And we're going from very low PMP22 levels in the cells up to very high PMP22 levels. And then what we've measured here is the trafficking efficiency of the PMP22 protein. And the trafficking efficiency is how much protein makes it out to the surface versus how much is trapped inside the cell. And so it's known that even under healthy conditions, PMP22 doesn't traffic to the cell surface with 100% efficiency. And what we see in our studies in these Schwann cells is as you go from low expression to high expression, the trafficking efficiency actually goes down from about 55% down to less than 30%. And so what this tells us is as you go to very high levels of expression, as takes place in the type 1A form of the disease, that less and less of the protein makes it out to the cell surface, and it instead accumulates inside the cell. Okay, and that actually makes sense as a disease mechanism. We wondered what happens to the protein that's trapped inside the cell. Does it accumulate there or does it get degraded? And so we simply looked inside the cells as a function of expression level to see what we could see. So here we are, we're going from low expression to high expression. And PMP22 is red. And you can see just barely at very low expression, you can just barely see a diffuse red of PMP22 being expressed in these cells. At higher levels of expression, you continue to see the diffuse red, but now you also see these punctae. And these punctae are either aggregates or we call them inclusions of accumulated PMP22. Now, these have been seen before by other labs, especially Lucia Notterpeck's, but also in the talk yesterday from Sansara, they also reported these. We actually know these are not in the endoplasmic reticulum. We've looked very hard. They're not there. We actually see them in, of all places, endosomes. We have no idea why they're in endosomes. But anyways, that's what we see. And so we wondered, is it these punctae that are driving the disease? And so we set out to try to figure out how to prove whether they were causing the disease. And all our experiments to prove it didn't work, but we stumbled on this accidentally. And the reason is because, so this is Jeffrey Lee, who's here and has a poster, and Catherine Stefanski, who also has a poster. And so we were interested in another mutant, the threonine 118 mutant of PMP22. And the reason we got interested in this mutant is it turns out that that 1% of all humans have this mutation. So some of us in the room, even who don't have diagnosed disease, probably have this mutation. And it had been reported in the literature to be a disease-causing mutation, but it was controversial. And so the fact that it's a 1 in 100 mutation tells us it can't be a definitive cause of the disease, because all the cases of Charcot-Marie Tooth disease are thought to be, 1 to 2,000 or so. And so this suggests that this is only a risk factor for the disease, that it predisposes you to Charcot-Marie Tooth, but it's not sufficient to cause the disease. And we wanted to know why that is. And so we did these same experiments on this protein that we did on the other disease mutants. And what we found, to our surprise, is that the protein is in fact unstable and it mistraffics. And yet it doesn't cause obvious type E Charcot-Marie Tooth disease in most people. And so why is that? Well, we look for it to form aggregates like all the other mutants do that cause Charcot-Marie Tooth disease. And we found that even relative to the wild type protein, it has a low propensity to form aggregates. So somehow the mutation makes the protein unstable, it makes the protein misfold, makes the protein lose function, but it doesn't form these punctae, and therefore it doesn't usually cause disease. And so that strongly implicates these punctae as the driver of the common type 1A form of the disease. And so that's pretty much what I just said, so I'll just move on. So basically what we think then, based on these results, is that under healthy conditions, you have sort of a Goldilocks situation, not too much PMP22 on the cell surface and not too little. Any misfolded, trapped PMP22 gets degraded instead of forming inclusions. And as a result, then you have normal, healthy myelination of PNS axons. Under these conditions, you probably have too much PMP22 at the cell surface. This could contribute to the disease. We suspect that it probably is a mild contributor to the disease, but I don't think anyone knows for sure. But we think that probably what the main driver of the disease are these intracellular inclusions that accumulate inside the cell, and that these somehow lead to myelin disruption of myelination and peripheral neuropathy. We know these are not toxic to the cells in the sense that they don't kill the Schwann cells. However, they probably interfere with we know they interfere with the ability of Schwann cells to myelinate. And that's not surprising because Schwann cells have a very complicated developmental program leading up to myelination. So what are the implications of this for CMT therapies? So we believe for the type 1A disease, what you would like, if you were going to make a drug to target this, is lower total expression of PMP22 and or compounds that prevent the formation of these intracellular inclusions. For the HNPP form of the disease, where you make too little of the protein, you would like a compound that increases expression and or increases the efficiency of folding and trafficking. And then for the type E mutations, for most of them at least, you would want to increase the efficiency of intracellular folding and cell trafficking. So over the last decade, my lab has developed a very robust assay for quantitating what happens to PMP22 when it's made in Schwann cells. And I won't go through the assay except to say what you get out of this assay is very quantitatively, with very tight statistics, the surface level of PMP22 that gets made, how much PMP22 is trapped intracellularly, the total PMP22, and the trafficking efficiency. And so this assay then we packaged into we reformatted this assay for a multi-well plate so that we could do high throughput screening using this assay to look for compounds that had the properties that I just described to you as possible therapeutic leads. And so what we've been able to complete recently with the support of the Charcot-Marie Tooth Research Foundation which actually I should point out, it actually started as an NINDS R21 grant that then was picked up by the CMTRF. So we've done I know for those of you who are real drug developers, a 20,000 compound screen is a little screen. For us, this was like Mount Everest. So we screened to our current 14 or so compounds that we're working with. And so this is just a little bit of the data of what these compounds actually do. So what all this is the surface level of PMP22 at the surface of these rat Schwann cells. And these are different compounds that we've discovered. And so these compounds increase surface trafficking. And so they would be of interest possibly as potential lead compounds that could be developed for the HNPP form of the disease caused by underexpression of Charcot-Marie of PMP22. And then these compounds all reduce surface trafficking. And so these would be of interest for either type E disease or the type 1A disease. And so this is really kind of where we're at with this project right now. This is very, I hesitate to even call this drug discovery because these are pre-lead compounds still. This is very, very early work. And so the next steps for us, so we've started to measure the EC50s to get some idea of potency. We have compounds in the range of 2 to 5 micromolar. We have other compounds that we've already dropped out of this because you have much higher EC50s. And where we're headed with this is we really need, the next step is to test the efficacy of these hits using a in vitro myelination assay. Bruce Carter is working on that. The difficulty of running these assays, I think, is a real limit to the whole field of people studying Charcot-Marie tooth disease. These are really, really difficult assays. And I refer you to the actual dental tooth poster out there as a really clever way of possibly finding the basis for a more efficient way of running these assays. And then we need, if we see efficacy in the myelination assays, we want to go on to mice, obviously. We still want to do additional high throughput screening to find additional compounds. And part of the reason for that is we don't believe that most of our compounds act by actually hitting PMP22 directly. So they're changing the trafficking of PMP22 indirectly. That's okay. One of them could turn out to be just the right compound, but everything's easier if you know upfront what target you're hitting. And so we have to do SAR to find derivatives of our initial hits that are better. And then actually we have another collaborator, Lars Plate at Vanderbilt, who, if we are working with compounds that don't hit PMP22, he has the proteomic tools that should allow us to determine what is the actual target that these compounds are hitting. And then we also want to test our hits with the CMT mutant forms of PMP22. And with that, I will stop. I just want to thank, again, the Charcot-Marie Tooth Research Foundation, NINDS. We do have an Ancora Deerfield project, which I didn't talk about today. I wrote an article about this project for the Protein Society newsletter that you can find at the Protein Society website if you're interested. And then I think I've mentioned most of our collaborators, and just want to thank you for your time.
Chuck was pretty efficient, so we have time for maybe one question before the general panel takes.
Just to help interpret the C118M results, have you tested trafficking effects of putatively benign variants at all?
Trafficking effects of benign variants.
So the idea is, of course, if you just grab a variant at random, it can have functional effects in these kinds of assays. So if you just take polymorphisms and test to see if they have trafficking effects of some kind, like you see for 118, then that would suggest the possibility that what you're seeing for 118 is incidental. So it's a good comparison to make. Just wondered if you've done that.
Well, of course, all our screening was not done with a disease mutant. It was done with wild type. And so
it's not taking the wild type, it's taking other polymorphisms that you don't think cause disease and see if they have any functional effects. Because of course, putatively benign variants will also have some often functional effects in these kinds of assays.
Yeah, no, we have not done that. So, yep.
Okay, thanks, Chuck.